Good morning, good afternoon, and good evening to everyone who joins us from around the world. We're so glad you're here.
It is wonderful to be back with you all.
This is Andrew and Renee. Welcome to the Stack series.
And we have a really fascinating topic lined up today.
Today, we are exploring the renaissance of ancient therapeutics and lost medical knowledge.
And, this is a topic that really challenges the way we look at time, at progress, and, frankly, human arrogance.
Right. We have this default view of medical history as this perfectly straight line. We think we started with, primitive superstition, mud, chanting, and then just marched steadily forward to the molecular precision we have today.
Exactly. It's the iPhone model of history, right?
Yeah.
The new version is always, by definition, better than the old one. Old equals bad, new equals scientific.
But what if that line isn't straight at all? What if it's actually a circle?
That is the central tension we're looking at today. Because right now, modern medicine is facing a genuine crisis that is forcing us to look backward.
And you're talking about the antimicrobial resistance crisis, AMR.
That's right. I mean, the numbers are just staggering. The sources we looked at mentioned that by 2019, nearly 5 million deaths were associated with bacterial antimicrobial resistance.
5 million. That's hard to even comprehend.
It's more than HIV, AIDS, and malaria combined in some years.
Yeah.
We are, and this is not an exaggeration, we are running out of modern weapons. The antibiotics that have kept us safe for the last 80 years, they're failing.
So scientists are being forced to do something that sounds, on the surface, completely counterintuitive.
They're going back, they're mining ancient texts, medieval leech books, battlefield journals from the 19th century.
But, and this is the critical part, they're not looking for folklore.
No, not at all. They're looking for hard data. They're essentially resurrecting lost cures, but they're doing it with some of the most advanced tools we have.
Like artificial intelligence.
AI, genomics, advanced chemistry. It's like Indiana Jones meets a biotech lab.
I love that. And the scope of what we're going to explore today is just massive. We're talking about everything from something called yellow soup,
Which is exactly what it sounds like.
to using AI to literally bring antibiotic molecules from Neanderthals back from extinction.
It really is a journey from the very, let's say, gross to the very high-tech. And quite often, they turn out to be the same thing.
So let's start unpacking this. Let's start with the foundations. I think we have to tackle this assumption that ancient healers were, just guessing, just throwing spaghetti at the wall.
That's the biggest misconception, and it's so important to get this right. We have to distinguish between theoretical medicine and what we call empirical medicine.
Okay.
Ancient healers didn't know why something worked. Germ theory didn't exist. They didn't know about DNA or receptors or anything like that. They had no microscopes.
But they knew that it worked.
They knew that it worked. Through centuries, sometimes millennia, of trial and error. That is data.
It's like the difference between knowing how to drive a car and knowing how an internal combustion engine works, right?
That's a perfect analogy.
You can drive to the grocery store perfectly well without understanding the physics of the pistons or the stoichiometry of the fuel injection. You just know that when you press this pedal, the car goes.
Precisely. And if a remedy killed the patient, the healer stopped using it. Or the healer lost their business. Or in some cases, their head.
Right. High stakes.
Very high stakes. If it worked, they wrote it down. And maybe the greatest example of this, the one that really proves the value of this data mining of the past, is the story of Youyou.
This is an incredible story. It reads like a Cold War spy thriller.
It really does. So to set the scene, you have to go back to 1967. The Vietnam War is raging, and it's a brutal conflict. But there was this third combatant that was killing everybody, malaria. It was absolutely decimating the soldiers on both sides, but especially the North Vietnamese forces fighting deep in the jungle.
And the really terrifying part was that their main drug, chloroquine, it wasn't working anymore.
The resistance was incredibly high. The parasite had evolved. The shield was broken. So the North Vietnamese government secretly reaches out to China for help.
And this is where the project begins.
Yes. Mao Zedong launches a secret military operation called Project 523. The 523 just stands for the date, May 23rd. And the mission was simple. Find a cure for malaria, or we could lose the war.
And they put a pharmaceutical chemist, Youyou, in charge of a team.
They did. And initially, her team did exactly what you would expect modern science to do. They didn't start with old dusty books.
They started with modern chemistry.
Right. They did what a Pfizer or a Merck would do today. They took thousands upon thousands of synthetic chemicals off the shelf and just threw them at the malaria parasite to see what would stick.
How many did they screen?
The sources say they screened 240,000 compounds.
240,000.
250,000 attempts.
And the result?
0. Nothing worked safely and effectively. It was a total crushing failure.
I just can't imagine the pressure. You're leading this secret military project. Soldiers are dying every day. The government is breathing down your neck, and you've failed nearly 250,000 times.
So, really out of desperation, Youyou pivots. She realizes the modern library is empty. So she goes to the ancient library.
Okay.
She starts digging through ancient Chinese medical texts, looking for any traditional remedies for fever.
And that's how she found the plant.
That's how she zeroed in on a plant called sweet wormwood, or Artemisia annua.
Mm-hmm.
She found it referenced in a text from the 4th century written by a scholar named Zhehong. The book was called The Handbook of Prescriptions for Emergency.
And he mentioned using this wormwood for fevers.
He did. He talked about using it for intermittent fevers, which is how they would have described the cycles of malaria back then.
But here's where it gets really fascinating, because initially it didn't work in the lab.
No, it didn't. They tested the wormwood extract using all the standard modern methods, which involved boiling it to concentrate it. And it failed. The efficacy was flat.
And most people would have just given up right there. They'd say, see, ancient folklore, just superstition.
But Youyou didn't give up. And this is the genius of her approach. She didn't assume the ancient book was wrong. She assumed her method was wrong.
So she went back to the source text.
She went back to the text. She reread Gabong's specific instructions. And the text said, soak it in 2 liters of water, wring out the juice, and drink it all.
Soak it.
Soak it.
Not boil it.
That was the aha moment.
No.
In traditional Chinese medicine, like when you're making tea, you almost always boil the herbs. You use heat to extract the compounds. But Gahong specifically said to soak it, implying cold water, to the Emelang. You realize that the heat from boiling was destroying the active ingredient.
That is such a critical detail. It's basically a chemistry instruction from 1,600 years ago.
It is.
It's like if you cook an egg, the proteins change. You can't uncook it. She was cooking the medicine to death.
Exactly. She realized the active compound was heat labile. It breaks down with heat. So she completely changed her method. She used a low-temperature ether extraction.
Ether.
Ether boils at about 35 degrees Celsius, which is cooler than your own body temperature.
Yeah.
So this allowed her to pull the chemical out of the plant without cooking it.
And when she tested that cold extract on mice and monkeys...
100% efficacy. Complete inhibition of the parasite.
Yeah, 100%.
It was a massive breakthrough. But the story gets even more dramatic from there. They needed to test it on humans to make sure it was safe.
But it's a secret military project. They can't wait for a formal, phase 1 trial protocol.
Time was everything. So Youyou volunteered to be the first human test subject.
She took the experimental drug herself.
She and 2 of her colleagues did. She put her life on the line for the data. She suffered no ill effects. So they moved immediately to clinical trials on the front lines in Vietnam, and the patients recovered with incredible speed. They isolated the compound, which we now know as artemisinin.
And today, artemisinin is the frontline treatment for malaria worldwide. It's saved millions of lives. And the key, the entire discovery, came from one sentence written in the 4th century.
It proves that these texts contain specific, replicable chemical instructions. It wasn't magic. It was a recipe for chemical extraction that we had just forgotten how to read.
Yeah.
It teaches us that observation over centuries is its own form of peer review.
No, she won the Nobel Prize for that, and rightly so. But there are other ancient therapies making a comeback, and some of them are a little harder to swallow, literally and figuratively.
Chemicals, yes.
Let's talk about the battlefield and bugs.
You were referring to maggot debridement therapy, or MDT.
I am. Maggots.
Right.
It triggers a very visceral yuck response in most people.
It does, and that yuck response is actually a huge part of the story. But if you look at the history, using fly larvae to clean wounds is a technique that pops up all over the world. Aboriginal medicine, Mayan culture.
And notably, in military history.
Yes. The sources point to Napoleon's surgeon, a man named Baron Dominique Jean Leray. During the Napoleonic Wars, he noticed something really strange.
What was that?
The soldiers who were left on the battlefield for days, hiding in the brush, their wounds infested with maggots. They actually had a better survival rate than the soldiers who were brought back and treated immediately by the doctors.
That's a total paradox. You see bugs in a wound, you think infection, rot, death.
Right? But Larry realized the maggots were acting as cleaners. Then we see it again in the American Civil War with a surgeon named John Forney Zacharias. And finally, it gets formalized in World War I by Dr. William Baer at Johns Hopkins.
And Baer's the one who really brought the scientific method to this.
He is. He figured out the key problem. While the maggots were cleaning the wounds, there was always a risk they could introduce other bacteria.
Like tetanus or gangrene.
Exactly, because they came from dirty flies. So he figured out a way to rear sterile maggots in a lab. That was the breakthrough for safety.
So let's break down the mechanism. Why is a maggot better than a surgeon with a scalpel? I mean, a surgeon is a highly trained professional. Why replace them with a larva?
It all comes down to precision. A surgeon trying to cut away dead tissue, which we call debridement, inevitably cuts some healthy tissue as well. It's just very hard to see the microscopic boundary between dead flesh and living flesh.
Okay.
But the larvae of one specific fly, the green bottlefly, Lucilia sericata, are extremely specific eaters.
They're picky eaters.
Very. They simply do not eat living tissue. Their mouthparts literally can't chew healthy flesh. Instead, they secrete these powerful enzymes that dissolve necrotic dead tissue into a kind of soup.
And then they just slurp it up.
They just slurp it up. It is microsurgery at a cellular level of precision.
And it's not just cleaning, right? They also disinfect the wound.
They do. They secrete antimicrobial peptides, which are basically natural antibiotics that kill bacteria, even superbugs like MRSA, that our modern drugs can't touch.
So they're cleaning, disinfecting, and I think I read they also promote healing.
They do. The physical action of them wiggling around stimulates blood flow and tissue recovery.
So we had this miracle cure, basically, in the 1930s. It was cheap, it was effective, it was safe. Why did it start?
Penicillin. As soon as we discovered antibiotics and germ theory really took hold, the idea of intentionally putting dirty bugs into a wound just became repulsive.
The yuck factor won.
It did. We traded biological complexity for chemical convenience. It was easier, cleaner, and felt more modern to just take a pill or give an injection.
But now, with things like diabetic ulcers and antibiotic-resistant infections on the rise, the maggots are coming back.
They are. The FDA approved them as a medical device back in 2004, which is interesting language, right? A device, not a drug. But again, the biggest barrier today isn't efficacy. It's that same squeamishness from both patients and doctors.
And speaking of squeamish, this feels like the perfect time to talk about yellow soup.
Yes.
This brings us back to Gehong again, the same 4th century scholar who gave us the malaria cure.
It seems he had a remedy for everything. In that same handbook, Gehong prescribed yellow soup for patients with severe food poisoning and diarrhea.
And yellow soup is, well, let's just say it.
It's a fecal slurry, a suspension of healthy human stool taken orally.
Which in modern medicine we call fecal microbiota transplantation, or FMT.
Exactly. And the underlying logic back then and the logic now is actually identical. You are restoring an ecosystem.
Explain that.
If you think of your gut as a complex forest, a severe infection, or more commonly, a heavy round of antibiotics, is like a forest fire, just burns everything down. The bad bacteria, sure, but also all the good bacteria that keep you healthy.
And when the forest is burned to the ground, the weeds take over.
Precisely. And the most dangerous weed in a modern hospital setting is a bacteria called C. difficile. It's a nasty pathogen. It causes severe, sometimes life-threatening diarrhea. And because it forms these tough little spores, it can hide. You can bomb the gut with antibiotics, you kill everything else, but the C. diff spores survive, and then they take over even more territory.
So the antibiotics often make the problem worse in the long run. They just keep burning the forest.
FMT is like replanting the forest. You introduce this entire diverse community of healthy bacteria from a donor, and they just outcompete the pathogen. They take up all the space and all the resources, and the C. diff basically starves.
And the success rates they're just staggering.
I mean, we are seeing cure rates well over 80%, sometimes up to 90% for recurrent C. difficile infections that have failed all other treatments. It was rediscovered in modern medicine in 1958 by Dr. Ben Eisman, who used fecal enemas. It's now becoming a standard of care for these specific, highly resistant infections.
So we've got plants, bugs, and poop. It really challenges this idea that high-tech is the only path to healing.
It really does. But this isn't just about dusting off old remedies. There is a very active, very complex debate happening right now about how to integrate these living biological therapies into a system that was designed for static chemicals.
Right.
And that brings us to the world of phage therapy.
Okay, let's unpack this. Phages. These are viruses, but they're the good guys.
Chuckles.
Yeah.
Well, it depends on your perspective. If you're a bacterium, they are definitely not the good guys.
Fair enough.
Bacteriophages, or phages for short, are viruses that only infect and kill bacteria. They are the natural predators of bacteria. They're the most abundant biological entities on Earth.
Yeah.
Wherever you find bacteria, you will find phages hunting them.
What do they look like?
If you see them under an electron microscope, they look like little lunar landers from an old sci-fi movie. They have a head, a tail, little leg-like fibers. They land on a bacterium, inject their DNA, and turn that bacterium into a factory to make more phages until it literally bursts. Boom. Now here's the thing. These were discovered over 100 years ago, and in the former Soviet Union and Eastern Europe, places like Georgia and Poland, phage therapy never went away. They've been using it continuously for decades.
But in the West, we abandon it.
We completely abandoned it for antibiotics.
Why? I mean, if it works, why did we stop?
Because antibiotics were so much easier. An antibiotic is like a carpet bomb or a nuke. It kills a broad spectrum of things. The pathogen, yes, but also a huge chunk of your good gut microbiome. But it's easy to prescribe. You don't always need to know exactly which bacteria it is. You just bomb the whole area.
And phages are different.
Phages are snipers. A specific phage kills a specific strain of bacteria. If you have a staph infection, you need a phage that is programmed to target that exact staph. If the bacteria mutates even slightly, the phage might not work anymore.
So if I get a staph infection, I need a completely custom-made treatment for my specific bug.
Correct. And that leads to the really big debate that we see in the sources. The debate between a la carte versus pret-a-porter.
Personalized versus ready-to-wear, like buying a suit off the rack versus getting one tailored.
That's the perfect analogy. The pret-a-porter, or ready-to-wear model, is what the pharmaceutical companies want. You create a cocktail of, say, 10 phages that covers 80% of known staph strains. You put it in a bottle, get it approved, and sell it at CVS.
It's scalable, it's easy to regulate, easy to ship, easy to sell.
Exactly. But the risk is that bacteria might evolve resistance to that specific cocktail, just like they did to antibiotics.
Right. So what's the other model? The a la carte.
The a la carte model is what they've been doing in places like the Youyou Institute in Georgia for decades. A patient comes in with a resistant infection. They take a sample of the bacteria from the wound, they grow it in a petri dish, and then they go to their massive library of phages and find one that kills that specific infection.
So they test it first.
They test it, and then they brew a fresh, custom treatment just for that patient. It's incredibly effective.
But I can already see the problem. It sounds like a complete nightmare for the FDA.
It is an operational and regulatory nightmare. Our entire system for approving drugs is built for static chemicals. Aspirin is aspirin. The molecule does not change. A bottle of aspirin today is the same as a bottle from 50 years ago.
But a phage is a living thing.
It was an evolving biological entity. And if you are making a custom medicine for every single patient, how on earth do you run a randomized, double-blind clinical trial on that?
You can't. The drug is different for every single person in the trial.
You can't. So how do you regulate it? And this is where some countries like Belgium have been very innovative. They're using something called the magistral preparation approach.
What's that?
They treat phages like a compounded prescription. You know, like when a pharmacist mixes a specific cream for you based on a doctor's note?
Okay. I see.
They decided to regulate the process, not the final product. They accredit the lab. They say, we trust your methods for hunting phages, we trust your purification process, and we trust how you match them to the bacteria. And therefore, whatever comes out of that trusted process is deemed safe for that one patient.
That's a huge shift in thinking. Regulating the process, not the molecule.
It's almost the only way that these kinds of living drugs can work in our current system. And there's another layer to this debate.
Which is?
Synergy versus antagonism. There's this big movement to make antibiotics great again by combining them with phages.
How does that work? If the antibiotics are failing, why keep using them?
Well, because bacteria are smart. They build these fortresses called biofilms. You can think of it like a slimy, impenetrable bunker that they all live inside.
And the antibiotics can't get through the slime.
They just bounce off.
Yeah.
But many phages have evolved enzymes that can chew through that slime to get to the bacteria inside.
So the phages are like the sappers in an army. They blow a hole in the fortress wall.
Exactly. They breach the wall, and then the antibiotic can rush in and finish the job. That's phage-antibiotic synergy.
But the sources also mention antagonism, that they can sometimes get in each other's way.
Yes. The order matters a great deal. Phages need active, dividing bacteria to replicate. The factory has to be running. If you hit the bacteria with an antibiotic first, and that antibiotic just stuns the bacteria so it stops dividing, the phage can't reproduce. The antibiotic has effectively turned off the lights in the factory, and the phage can't work.
So they literally cancel each other out.
They can. So it's not as simple as just throwing everything in the infection at once. It requires a really sophisticated understanding of the biology. You might need to give the phage first, wait 6 hours, and then give the antibiotic.
This complexity is fascinating, but it also really highlights why Big Pharma has been so hesitant. It's hard to patent, it's hard to scale, it's a nightmare to regulate.
Which is the perfect bridge to the frontiers. We've looked at what we know and the debates that are happening now, but what might be coming next is, honestly, it's mind-blowing. We are talking about using AI to essentially time travel.
Okay, this is the field of molecular de-extinction. This is where we stop looking at just old books and start looking at old DNA.
Yes. This is the work of visionaries like César de la Fuente, working with data from people like Svante Pääbo, who won the Nobel for sequencing the Neanderthal genome.
So explain this to me. How do you find a drug inside a Neanderthal? It's not like they had pharmacies.
No, but they had immune systems. And every living thing, including us, produces peptides, small proteins that are a first line of defense against bacteria.
Our natural defense.
It is. And over millions of years of evolution, different animals evolved different peptides to fight the specific bacteria in their environment.
And the code for those peptides is written in their DNA.
Exactly. So what De La Fuente and his team are doing is using artificial intelligence to scan the genomes of extinct organisms. We're talking about Neanderthals, Denisovans, woolly mammoths, ancient elephants.
But the genome is huge. It's billions of letters long. How does the AI know what to look for?
That's the beauty of it. It's all about pattern recognition. The AI has been trained on what modern antimicrobial peptides look like. They tend to have a certain chemical structure, a certain shape, or a positive charge that lets them punch holes in bacterial membranes.
Okay.
So the AI scans the ancient DNA looking for these encrypted sequences that match that known pattern.
It's like a metal detector for weapons, but it's scanning ancient genetic code.
That's a great way to put it.
Yeah.
And when the AI flags a sequence, say, from a Neanderthal genome, the scientists can then synthesize it. They essentially print the molecule in a lab using chemistry. They don't need a live Neanderthal, they just need the code.
Did it work?
It worked. They found a molecule from the Neanderthal genome that they dubbed Neanderthalin. They synthesized it, tested it on modern pathogens in mice, and found it was effective against skin infections.
That is just incredible. A molecule that has not existed on Earth for 40,000 years is killing modern superbugs in a lab in 2024.
And they found another one from a woolly mammoth they called Mammathusin. The AI allows them to compress evolutionary time. De La Fuente himself estimates that the AI has accelerated this discovery process by 1 million years of research time.
1 million years.
Think about it. Evolution takes millions of years of trial and error to design a new defense molecule. The AI can scan the entire history of that evolution, the global hard drive of life in an afternoon. We are, in a very real sense, waking up ghosts to fight our modern battles.
And it's not just extinct animals. There's also the Ancient Biotics project at the University of Nottingham. This one really caught my eye because it involves a brass pot.
Yes, this is a fascinating return to that recipe concept we saw with Youyou. They took a other 1,000-year-old remedy for eye infections from an Anglo-Saxon text called Bald's Leech Book.
And the recipe sounds like a complete witch's brew.
It absolutely does. It calls for garlic, onions or leeks, wine, and bovine bile.
Cow gall. Delightful.
And here is the kicker, the really crucial detail. It must be brewed in a brass vessel and left to stand for 9 days.
Now, I feel like most modern scientists looking at that would just roll their eyes. Cow bile and brass vessels. It just sounds like pure superstition.
They were skeptical. But they decided to rebuild the recipe exactly as written. And what they found points to a crucial insight. Synergy is the secret.
What do you mean by that?
Well, they tested all the individual ingredients. Garlic contains allicin, which is known to be antimicrobial, but on its own. It didn't do much against the superbug they were testing. The bile on its own, nothing. The mixture without the brass vessel, a minimal effect.
But when they put it all together in the brass pot, for 9 days...
It annihilated MRSA biofilms. It killed up to 90% of this incredibly tough superbug.
So why the brass vessel? What's the magic there?
It's pure chemistry. The brass slowly releases copper salts into the mixture over those 9 days. The acidity of the wine and the bile helps to leach the copper out of the metal. And that copper then interacts with the sulfur compounds from the garlic and the bile to create entirely new chemical compounds that didn't exist in any of the raw ingredients.
So the pot is actually an ingredient.
The pot is a chemical reactor. The copper is acting as a catalyst. And this completely challenges the silver bullet method of modern drug discovery.
Right. Modern pharma is always looking for that one active molecule, one single chemical.
But these ancient remedies, they rely on synergy, a complex soup of chemicals all working together. It's the difference between a sniper and a shrapnel grenade. Resistance is much harder for a bacterium to develop against a complex soup. If you're being attacked from 5 different angles at once, cell wall, protein synthesis, DNA replication, you can't evolve a defense against all of them simultaneously.
We disregarded these recipes because they were messy.
And the mess might be the entire point. It might be exactly why they work.
Now, we're also seeing this blend of high-tech and traditional in other ways. We talked about the difficulty of manufacturing and shipping phages, but what if you could just email them?
This is the concept of the phage printer. It relies on synthetic biology. Instead of having to grow phages in these big, messy vats of bacteria, which is risky, you can use something called cell-free synthesis. All you need is the DNA code.
So the vision here is a patient walks into a hospital in, say, rural Idaho. They have a resistant infection. The doctor sequences the bacteria's DNA, and AI designs a custom phage to kill it. And machine in the corner literally prints that phage on the spot.
That is the ultimate vision. No more shipping samples to a specialized lab in another country. No more waiting for weeks. It transforms the phage from a product that you buy into a service that you access. You're not selling the drug, you're selling the digital code and the ink.
That sounds incredible, but it also sounds like it brings up a whole new world of complications, which I think leads us directly to the implications. We have to talk about the money.
We do, because the economics of antibiotics are fundamentally broken. It's a classic market failure.
Why is that? You'd think we all need them. You'd think the company that finally cures the superbug would make billions of dollars.
You would think so. But from a business perspective, antibiotics are terrible products. If you invent a new heart medication or a statin for cholesterol, the patient takes it every single day for 30 years.
Right. It's a recurring revenue stream, a
Yeah.
subscription.
Exactly. If you invent a new antibiotic, the patient takes it for 7 days, they're cured, and you lose your customer.
And what's more, you ideally want to use that powerful new antibiotic as little as possible to prevent resistance from developing.
That's the real killer. The best new antibiotics should sit on a shelf in a glass box that says, break in case of emergency. So the drug company spends $1 billion developing it, gets it approved, and then public health officials rightly say, great job, now lock it up and don't sell it unless the patient is about to die.
That is a terrible business model.
It's an impossible business model.
Yeah.
And that's why almost all of Big Pharma, the Pfizers, the Novartises of the world, have abandoned the field. It's all left to small biotech companies who very often go bankrupt trying to get a drug approved. The sources call this the valley of death in funding.
So if the market is broken, how do we fix the plumbing? Because we absolutely need these drugs.
The consensus is that we have to start treating antimicrobials like public infrastructure, like roads or bridges or fire stations.
Fire stations.
Think about it. We pay for the fire station to exist and for the firefighters to be ready, even if there isn't a fire today. We don't pay them per fire. We pay for the capacity to fight fire when we need it.
That makes perfect sense.
And that's the model being proposed by groups like CariBX and Incate. It's a subscription model for drugs. Governments would pay a guaranteed fee to the company just to keep that new antibiotic available, regardless of how many pills are actually sold. It completely delinks profit from sales volume.
There's also a major ethical question here. We're talking about mining ancient texts and indigenous knowledge. Who owns that?
This is the massive issue of biopiracy. If an AI in Silicon Valley scans a database of Ayurvedic medicine knowledge developed by Indian sages over thousands of years and finds a cure, who gets the patent? The tech company, or the culture that developed the remedy in the first place?
The sources mentioned something called the Traditional Knowledge Digital Library, the TKDL.
Yes, the government of India has been very proactive here. They are painstakingly documenting all of their traditional medical knowledge in a digital searchable format, specifically to prevent foreign companies from patenting things that have been public knowledge in India for centuries.
But it gets even trickier with this de-extinction stuff. Can you patent a molecule that you found in a mammoth?
It's a huge legal gray area. It's not natural in the sense that it doesn't exist anymore in nature, but it wasn't invented by the scientist, it was discovered. It's opening up a completely new frontier in patent law.
And then there's the question of access. If we do develop these incredible high-tech, AI-printed cures, will they only be available in wealthy hospitals in Boston and Geneva?
That is the enormous risk. The deep irony is that traditional medicine is often the only medicine available to the rural poor in the global south. If we extract the active ingredient, turn it into an expensive pill, and patent it, we might be stripping a culture of its own heritage and then selling it back to them at a price they can never afford.
That is a very sobering thought. And it feels like we're also seeing a deeper philosophical shift happening here, aren't we? For the last century, the metaphor has always been war. The war on microbes.
Yes, eradication, sterilization,
Mm.
killing. We viewed microbes as the enemy. But the success of things like FMT and phages suggests a completely different metaphor.
Which is?
Ecological management.
So we're not trying to kill everything, we're just trying to restore balance.
Exactly. We're finally realizing that the human body isn't a sterile fortress to be defended. It's a complex garden to be tended. Sometimes you need to pull a weed, that's the antibiotic. But you also need to fertilize the soil and plant good seeds, that's the probiotic or the FMT.
The old ways, which were always holistic, always synergistic, they weren't wrong, they were just unexplained.
And now we finally have the molecular tools to explain them. We aren't really going backward, we're spiraling upward. We're circling back to the wisdom of the past, but we're armed with the tools of the future.
And that combination seems to be our best hope against the superbugs.
It really is.
Before we wrap up, thank you to all our partners, sponsors, and scholars who made this podcast and the research possible. We're truly grateful for your support.
And to our listeners, thank you for being part of this journey with us. If you enjoyed today's episode, be sure to subscribe and follow Stack Studios for more informative content, and support us in Patreon.
Until next time, stay curious, keep learning, and let the search for wisdom guide you forward. Have a great day.